Hello, welcome back. This is Professor Young, and this is the beginning of our look at cell biology. And primarily, our main focus is going to be on the cell membrane. This will be part 1 of 2 for our WIMBA. And during this first part, we're going to be really focused on the overall structure of the cell membrane and begin to look at some of the ad hoc, if you would, or additional structures that are associated with the cell and the cell membrane. And so with that said, let's do a little quick review of the overall structure of a cell membrane. We should now understand that there are really 2 key aspects of a cell membrane. We have, I'm going to divide it right here along that line there. From that line down, we should understand that we have a hydrophobic area. It is made up of a glycerol group as well as 2 fatty acids. And you can see those 2 fatty acid tails that are hanging off of that glycerol group right there. And in addition to that, we also have from that red line up, we have a hydrophilic area. And that hydrophilic area is composed of a phosphate group as well as a choline group. And basically, choline is designed to enhance cell communication and a cell's ability to be able to effectively communicate between other parts of the cell membrane. And so that is your basic overall structure of the cell membrane. Now, one additional thing I will throw out here is this top area right there is very often referred to as the head, while the bottom portion right down here is very often referred to as the tail. And so they have a hydrophobic tail and a hydrophilic head. Now, why is this critical and important? Well, it comes down to a couple different things here. First thing, we should understand through our look at diffusion and osmosis within the lab that the cell membrane is a selectively permeable structure. The cell membrane is a selectively permeable structure. And when we say that, what we are actually meaning is that the membrane has the ability to regulate what enters and what leaves the cell. And that is what we refer to, that definition right there is what we are referring to as being selectively permeable. And so that selectively permeable membrane, also referred to as osmolemma, is made up of a repeating series of these phospholipids. And so that's what you're looking at right there. You're looking at that hydrophilic head and hydrophobic tail. Those 2 parts there make up a phospholipid. And so you've got a phospholipid attached to a phospholipid next to a phospholipid next to a phospholipid, 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 phospholipid. The story or the plot thickens a little bit more. And what we mean by a bilayer simply is that there are really a double layer of these phospholipids. And the way that these phospholipids are arranged in this bilayer is tail to tail. And so the tail of one phospholipid is butted up against the tail of another phospholipid that is directly underneath of it. And this bilayer arrangement goes all the way around the entire cell membrane. One of the other key aspects or key components of this structure, besides the fact that it is selectively permeable, it can regulate what goes in and out, besides the fact that it's got this unique setup where the external regions are hydrophilic and the internal regions, if you would, when the tails are butted up against each other, are hydrophobic. We also have the dynamic that this is a structure that remains completely flexible. There is fluidity, as we say, or movement that happens within the membrane. It is not a concrete, immovable structure. There is give that is built into this cell membrane. And we'll talk about why that might be here in a couple minutes. All right. Going back to this idea of being a phospholipid bilayer. Going back to this idea of being a phospholipid bilayer. Remember I said that the heads are arranged so that they face outward. Here are the heads. Here are the heads. And again, as I've already mentioned, these heads are oriented in such a way so that they face outward. They are facing outward. Right. That's number one. Number two, I also made mention that the tails are arranged in such a way that they face inward. And I think you can see that right there. And so you've got to kind of imagine that this basic structure, this phospholipid bilayer, is going all the way around the entire cell structure. It is making up the entire cell membrane. And as it's doing that, you've got this depth that is happening now within the membrane. It's not just a flat little line that is creating the membrane. It's this dynamic structure that has these hydrophilic heads. And because it's hydrophilic, because it's water-loving, this is where it gets a little deep. These hydrophilic heads are polar. That means that they're going to interact with water. And that's critical in how the cell membrane is able to be selectively permeable. These heads need to be polar. They need to interact with the environment that is directly in contact with them. If this is the external area right here, if this is external, you've got interstitial fluid that is over 90% water that is coming in contact with those heads. They need to be polar. They need to be able to interact and be hydrophilic with that phosphate group and that choline group. The same thing is true for here, the opposite end, where you also have those polar heads. Right here is inside of the cell. That's inside the cell. You've got cytoplasm that is swirling around inside of this cell. That cytoplasm is once again mainly made up of water. And so if you're going to have a structure that is coming in direct contact 24/7 with this cytoplasm and the interstitial fluid that is over 90% water, then you need to be able to have it be polarized so that it can interact freely with it and not repel it. Otherwise, you're asking for complete chaos. On the inside of that cell membrane, where the lipids are, the lipid tails are, we have a nonpolar region. We have a nonpolar region. These guys do not play nicely with water. They are lipid soluble. They interact freely with lipids, but not water. And that's okay in this case, because guess what? There's that thin layer of the in-between that's not going to freely be interacting with the water. And so there is no water, or I should say there isn't much water that's going to interact with those nonpolar areas. Now, this does set up a predicament for things that want to pass through. Because we've got to be able to create some kind of system where a lipid can still get through the polar heads, or where a water-based substance can still it's going to be able to get through the heads with no problem, but it needs to be able to pass through and get through these nonpolar tails in some way. And that's where the complexity deepens. Because we do have some additional structures that we need to pay homage to. We have these guys known as integral proteins and peripheral proteins. Integral proteins are the key for things to be able to pass through a cell membrane, despite whether it's lipid soluble or water soluble, because these integral proteins integrate. Integral proteins integrate themselves into the membrane. They extend from one side all the way through to the next side. And because they are in touch with both the external and the internal sides of the membrane, they can freely move things that are lipid and or water soluble without any kind of hesitation, without any kind of obstruction. But, there's always a but. Integral proteins also now have the same issues that everything else has. These integral proteins need to be able to freely interact and not repel a hydrophilic head and a hydrophobic tail. And the way that it does that is, and here's a fancy term for you, integral proteins hold on one second. Or what we refer to as being amphipathic. And amphipathic simply means that the structure that you're dealing with, in this case, an integral protein, contains, check this out, 3 distinct regions. It contains a hydrophilic region here and a hydrophilic region there. And it's also going to contain a hydrophobic region there. And so the integral protein itself has to be able to have different regions that are able to interact with the varying degrees of solubility found within that membrane. Now, peripheral proteins, the other major structure that we have associated with the cell membrane, doesn't have to worry about that. Peripheral proteins are strictly going to be hydrophilic because they're only going to be interacting with either the internal or the external sides of the phospholipid bilayer. In other words, it's only going to come in contact with the choline and the phosphate group, the water-soluble aspects of it. And so there's a lot less complexity there with the peripheral proteins. Doesn't mean that they're not as important. They are. It's very often with your peripheral proteins where enzymes are going to have the greatest influence and impact on the cell. And so, yes, you're going to have complexity and importance still within that peripheral protein. It's just going to be different than those integral proteins. And so here's what it looks like. Right now, we did this in lecture. I can't run it here because Women Classroom doesn't run animation. But I want you to think of this whole process as kind of being our swimming pool with our tennis balls, and we have our little rubber ducky inner tube, and we have our floating mat. Right? Whereas of course, the tennis balls are the cell membrane. The rubber ducky one second. The rubber ducky is the integral protein, and the floating mat, of course, would become the peripheral protein. And so there's our three key players as of right now. Then comes in the bowling ball, which is going to be some kind of disturbance that's going to happen within our cell. And so it comes in, it launches itself through the membrane, such as this, right? And it's going to cause everything from here over to go ahead and shift its way that way. And then once all the waves calm back down, it will go ahead and play itself back out and redistribute itself. It may not be in that same area again. All right? In fact, especially these integral proteins have the ability to wander in between all of these phospholipids. That's the beauty of the cell membrane. It's got flexibility. That's why we refer to it as, and here's like the Pee Wee Herman word of the day, if there was one, the fluid mosaic model. It's right here. Right? The fluid mosaic model. If we break this down, a model is something that's going to represent how something actually works or looks. Mosaic, because think about what a mosaic is. It's a bunch of glass or it's a bunch of tiles that have no relation to one another, but yet come together to form a picture. It's exactly what we have here. This is considered mosaic. This is considered mosaic because you've got individual pieces that are not directly tied to one another. The phospholipid bilayer and the integral protein and the peripheral protein are not anchored to one another. They are free to move about, but yet they still make up one structure. All right? And fluid, because there is movement, there is freedom to disperse. And so we think of the cell membrane as this fluid mosaic model because it is made up of independent parts that are free to interact with one another to create this dynamic structure known as the cell membrane that has the ability to regulate what is coming in and what is going out. If this was a static structure, and when I mean static, I mean non-movable, non-fluid based, you would lose the ability to some degree for this to be selectively permeable. And so it's critical that, yes, we have these structures that are here, but that they remained independent of one another and yet still interact with one another. Does that make sense? Hopefully. Hopefully it does. Now, let's get into where things start to really get interesting. All right. We understand the parts of being selectively permeable, or the parts of being a membrane. But how exactly do we control what goes in and what goes out? Well, in lab, and I'm not going to belabor this too long here, because I think in lab we've kind of done a sufficient job of going through and looking at what is diffusion versus what is osmosis. One of the things we will do here, though, is take it a step further and look at some more parts that make up diffusion, such as, and not limited to, facilitated diffusion and active transport. In general, when we take diffusion as just being diffusion, the movement of molecules from an area of higher concentration to an area of lower concentration, we understand that diffusion is a passive process. We understand that diffusion is a passive process. Meaning that it does not take in any energy. Osmosis is a special type of diffusion where we're not dealing with the movement of molecules, we're dealing specifically with the movement of water. Facilitated diffusion, so let me just kind of come back in here real quick. Osmosis is specifically the movement of water, right? Whereas facilitated diffusion is the movement of molecules with help. And active transport is the movement of molecules through the use of ATP. And so, in other words, active transport is energy consuming, whereas diffusion, osmosis, and facilitated diffusion tends not to be energy consuming. It happens passively. It happens passively. And so osmosis was the first one of those types of special cases of diffusion. And as we remember, or as we should recall, that we had these three basic scenarios by which osmosis will occur. And I don't think I threw this word at you earlier. I'm going to throw it at you now. It all revolves around tonicity. And tonicity really has to do with the ability and direction by which water will move. Sorry about that. I had to do that kind of broken here. And so tonicity is really all about the way at which water will move. And there's three scenarios by which water will move. We know that we can have hypertonic solutions. Right? We know that we can have hypertonic solutions in which there is more solute outside of the cell than inside of the cell. Keep in mind the definition of osmosis. Do not ever forget this definition. The movement of water from an area of higher solute concentration to an area of lower solute concentration. Right? That's your definition of osmosis. Water is always going to move from where there is less to be dissolved to an area of where there is more to be dissolved. And so in a hypertonic solution, what we are implying is that there is a greater solute concentration in the extracellular, or I should say the intracellular material outside of the cell than what there is in the than what there is inside of the cell. 0.9% sodium chloride in the cell, 10% sodium chloride out of the cell. The water is going to move out, which is going to cause shrinking within the cell. Crenation. Crenation. Hypotonic solutions, on the other hand, we see a reversal where there is less water. Where there is less solute concentration outside of the cell, and where there is more on the inside. And so in this case, if you've got 0% sodium chloride on the outside, you've got 0.9% on the inside, water is going to want to move into the cell. And that creates that hypotonic environment. Right now, very often this leads to cell rupture. This leads to a rupturing of the cell membrane, what we call lysis, right, or a lytic state. And then we have good old isotonic environments. Right? An isotonic environment, the external environment is going to be equal to an internal environment. 0.9 on the outside, 0.9 on the inside. There is no net gain in the water movement. Everything has reached equilibrium. And so that's your basis between hypo, and isotonic solutions. Now, facilitated diffusion, which is something that we have not discussed to date, is a little bit different. All right, and actually, before I do this, let me just kind of come back, and I'm going to jump ahead one slide here and look at types of membrane proteins, because this is going to set us up for both facilitated diffusion and active transport. Right? There are 4 generalized types of proteins that are going to be associated with the membrane of a cell. We've got structural proteins and transporters, right? And both of these are what we are going to refer to as, oh, that's a horrible color. Hold on one second. Both of these are going to be as to what we refer to as integral proteins. And then we also have enzymes. Oops, try that again. We also have enzymes and receptors, which are going to be more commonly referred to as peripheral proteins. Peripheral proteins. We are more so going to be concerned with, at least for the time being, we're going to be more concerned with secondarily the structural proteins, because that's really where tissue types are going to be played out, and that battle is going to be fought. And for right now, transporters, because we have two types of transporters. We have two types of integral proteins that are going to move substances in and out of the cell. As you have probably guessed, carrier proteins and channel proteins are the two that we need to look at. Your carrier proteins are typically always associated with facilitated diffusion. Your carrier proteins are almost always associated with being some type of facilitated diffusion. Now, notice the definition of facilitated diffusion: the movement of a molecule from an area of higher concentration to an area of lower concentration. Get ready for it. This is the biggie. Via a carrier protein. I cannot stress these last four words enough. I can highlight it. I can scribble on it. I can put a box around it. I can put arrows all around it, go underneath of it. I can do whatever I want to do. You need to make sure that you understand that the difference between facilitated diffusion and diffusion is the fact that facilitated diffusion has facilitation. It can facilitate. It can help. It helps diffusion occur with the process of a carrier protein. If we go back to the pool sample, our little rubber ducky inner tube becomes my carrier protein. And that carrier protein is going to be specific for certain types of ions or molecules. Not just anything is going to be able to pass into the cell via a carrier protein. Because my little rubber ducky inner tube that's acting as my integral protein known as a carrier protein is a bird, ducks are birds, it is only going to move birds into the swimming pool. It's not going to move frogs in. It's not going to move bats in. It's not going to, actually, I stand corrected. It may move a bat in. I'll explain why in a couple seconds. It's not going to move a grasshopper in. It's not going to move toads in. It's not going to move snakes in. It's not going to move people in. Because my carrier protein is a bird, it is only going to move birds into the pool. It's selective. There needs to be a fit in order for it to happen. And once the fit has happened, then yes, it will bring the bird or the ion or the molecule into the pool or into the cell. Now, I kind of hinted or alluded at the fact that maybe, just maybe, a bat could get into my pool using my rubber ducky inner tube as its carrier protein. And the reason why that happens is because even within the cell, even with a high degree of specificity, there's a term that we heard when we talked about enzymes. We know that enzymes work off of specificity. Carrier proteins, same thing. A bird can fly. A bat can fly as well. Maybe that is enough closeness in specificity that the bat would be able to sneak in. An ion that was this shape and an ion that was that. These are two totally different things. These guys would never be able to use the same carrier protein to enter into an area. But, how about something that would look like this? That purple one is a combination of the two blue ones up there. And so therefore, that purple molecule could use a carrier protein that was designed for the blue rectangle because it's got a similar shape to it. Or it could use a carrier protein that was had a high degree of specificity for being circular because it's got a circular end to it as well. So while the blue square could never use a carrier protein that the blue circle uses, and vice versa, the purple square circle combo could indeed use a blue square carrier protein or a blue circle carrier protein, as is the same case for why a bat, which flies, could possibly use a rubber ducky inner tube as its carrier protein to get into the pool. Now, I know that's a little out there. Right? And so, what does this look like in actuality? Well, before we go down that avenue, let's take 2 minutes and talk about exactly how carrier proteins do what they do. All right? I'm going to go back to the fact that we have a high degree of specificity. The carrier protein is very specific for only a particular molecule or a close relative, the bird or the bat. And what ends up happening here is that molecule needs to be able to fit into a trigger zone within the protein that will allow it to open and close. These are gates that are highly selective. They are highly selective. I will say it again. They are highly selective. They are highly selective. Because they are highly selective, they cannot be open all the time. And so these are going to be gateways that are only going to be open in the presence of a very specific molecule. Because of this selectivity, because of the high specificity for particular molecules, this is a very slow process that occurs across the membrane. 1,000 to 1,000,000 molecules per second. Now that may sound fast, but in the terms of physiological response, that is not. Let's take a look at how exactly this happens. For facilitated diffusion to occur, all right, first thing, here's our molecule. All right, we will notice that the gate is open. We will also notice here's the bonding receptor that molecule needs to be able to fit into. If that molecule does not fit into that barrier, carrier receptor, it's a no-go. Right? Notice on the back end here. Notice on the back end that gate is closed. There's no way that molecule is going to get in. Now, let's fast forward. That molecule that is right here does enter in, and it does fit. It does fit into that binding receptor. And so what ends up happening is the gate closes. And here you can see the gate is closed. And what ends up happening is the gate will open right there. Once that gate is open, then that carrier protein will go ahead and will release the molecule in. Now, this can be a bidirectional process, and so what could very well happen is another molecule that is sitting here, right there, could very easily go ahead and enter back in and bind in, and then reverse the process once that gate is open to leave. Right? And so this can be a bidirectional process. But for all intents and purposes, this is how facilitated diffusion works. Now, we are going to stop here for this WIMBA session. I'm going to record a part 2 to this, which is going to have us looking at channel proteins. It's also going to have us really taking a look at a little bit at active transport and the cytoskeleton. What do structural proteins look like? What kind of structural proteins are there? And how do they relate to what we've been talking about in the lab? Epithelial tissues and connective tissues, as well as muscular tissue and nervous tissue and glial tissue, which we haven't got to yet, but we will. We will. And so with that note, I'm going to let you go and have some fun studying and reviewing. If you have any questions, as always, I cannot stress it enough, please, 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 please feel free to email me, ask me those questions. I will get back to you. And on that note, have a good day, and I'll see you in the lab or lecture.